So let's start the session. So in this session, today we'll be learning about the biochemistry subject and we'll be discussing the previous year question paper, which is of the year 2022. So before starting this, I would like to say, don't worry that the JNTU paper is finished. So our main motto for conducting this session is, as we know that the OU and the AU university candidates are going to write examinations. So we have conducted this session to help the candidates who are going to write their examinations. And I hope this session will be very useful for you people. And do follow us and stay till the end. And this, I will guarantee you that this session will be very interesting and very helpful for you all. So please do stay till the end. So before start, let's start with 2 mark answers, which are short answers. So this is the first question what we have got in the last year. The question goes, what are the functions of lipids in the human body? So can you say what are lipids? Lipids are triglycerides. So what are their function? What does they do in our body? How do they help in our to our body? So here are the functions. They help in fatty liver prevention, which means that they don't make the liver accumulation of fats on the liver. They transport cholesterol and form lipoproteins. They constantly supply fatty acids for synthesis of eicosanoids. Eicosanoids are nothing but they are lipid-based signalings which help in unique way and give in different response. They also promote and prolong clotting time and increase fibrinolytic activity. So the daily intake of fats in our meal will also reduce atherosclerosis. Next question, define redox potential. What is redox potential? It is a reduction and oxidation potential, which means it calculates the oxidation and reduction potential. It is also represented as EH, and it measures the tendency of chemical species to gain or lose electrons. Okay. What are isoenzymes and allosteric enzymes? This is a very good question, and this question comes from the part of unit enzymes. And this is the most repeated questions, and you must focus on this question. And mostly it might come as a long answer, as well as mostly it repeats on for short answer question for 2 marks. Okay, so please be focused on this question. Okay, isoenzyme. Isoenzymes, they're also known as isozymes. So enzymes, as we know, there are different forms of enzymes, and these enzymes help in catalyzation of reactions. As you know, there are different enzymes that catalyze, but these isoenzymes, what they do is they have different properties. So what they have, they help in, and the specific property of these isoenzymes is they help in tissue distribution. They're typically encoded, means these enzymes are encoded by different kinds of genes. As we know that we are having so many types of genes, right? So, and they also do post-translational modifications. In our gene, we know there are different modifications, and that's what modifications make species different, right? So these enzymes make modification on our genes. And the allosteric enzymes. Allosteric enzymes are the enzymes that have an additional binding. As you know, every enzyme has a binding site, okay? So allosteric enzymes, what they have is, they have an additional binding site. So whatever and the enzymes come and bind, they bind from the regular site as well as they have an additional binding site where the molecules can bind and activate. Next question. Mention types of RNA and their function. There are majorly three different types of RNAs: messenger RNA, transfer RNA, ribosomal RNA. So the cellular composition of messenger RNA is 5 to 10%. The transfer RNA is 10 to 20%. The ribosomal RNA is 50 to 80%. So besides these three, there are also additional RNAs, which are also present in our cells. They are heterogeneous nuclear RNA, which is known as hnRNA, small nuclear RNA, snRNA, and small nuclear RNA. So see the difference. That is small nuclear RNA, and this is nuclear RNA. And the other is small cytoplasmic RNA, which indicates scRNA. So what is messenger RNA? As the name indicates, messenger means it provides a template for protein synthesis. And transfer RNA, what does it do? It transfers the genetic code and brings near amino acids. And these amino acids read the genetic code and identify the code, and they do their action. Ribosomal RNA, they play in structure and catalytic role during translation. Next question. Define endergonic and exogonic reactions. What are exogenic reactions? These have free energy, and that means these reactions can occur without energy. As you all know, the energy symbol is delta G, and in exogenic reactions, the delta G is negative. So that's what we call exogenic, means excluding the energy. Okay. So what happens is the exogenic molecules have spontaneous reactions. So as they are not using any energy. And endergonic reactions. These reactions are delta G positive, which means whenever these reactions are occurring, they need energy. And the delta G will be positive here. These reactions are non-spontaneous because whenever they need energy, right? So whenever energy they are using, so there will be a little time period for them. So they do not occur continuously because it happens. They need their energy, what is coming is utilized, and the energy what it is, again after utilization of energy, again they have to take another energy. So like that, it is not continuous process. Okay, they need energy. Next question, write a note on tyrosinemia. Tyrosinemia is a rare and genetic disorder characterized by body inability to break down aminotyrosin. So if our body is not breaking down the amino acid, and if the amino acid gets accumulated in our body, the person might suffer from tyrosinemia. So whenever these amino acid, anything that is getting accumulated, it becomes toxic. And that toxicity leads to different kinds of health problems. So to identify a person is suffering from tyrosinemia, you have every disease has symptoms. So likewise, tyrosinemia is also having symptoms. So what are its symptoms? Bloody stools, and the person is smelling cabbage-like odor. He's having frequent diarrhea. He suffers from fatigue. He's losing his weight and is having continuous vomitings. So when a person is having these symptoms, you can identify him as tyrosinemia person. Okay. So the treatment usually we use for treating tyrosinemia is either you would improve your diet, or if it is a severe case, there will be a liver transplantation. Okay. So next question. Explain the biological significance of ATP and cyclic AMP. So can you just write down what is the full form of ATP and cyclic AMP? Anyone? Okay. So ATP is adenosine triphosphate and cyclic AMP is adenosine monophosphate. So ATP is an energy. ATP acts as an energy in cells. Means whenever a person is doing any activity like you're doing running or jogging or any other work you're doing, you need energy, right? So that energy is your ATP. So every cell to perform any task, they need energy. So our energy is in the form of ATP, right? So these ATP help in exergonic reactions. So as we have discussed earlier, endergonic means does not need energy, and exergonic does need energy, right? So these ATP help in our exergonic reactions or hydrolyzing any higher compounds to hydrolate a molecule or dehydrate molecule. To hydrolate it, we need ATP. It can also supply energy to endergonic reactions by converting them to its phosphate form, like adenosine diphosphate to phosphate. By hydrolysis, what happens here is adenosine monophosphate converts to phosphate, and the bond formation happens from adenosine diphosphate to monophosphate. Okay. Cyclic AMP, or you can call it as CAMP pathway. What is the significance is they act as a secondary messenger. They use an intracellular signal transduction, which this intracellular signal transduction involves in transferring of cells. Okay, and these CAMP binds to ion channels like hydrogen cyanide and regulates passage of specific ions across the membrane. So what happens, like whenever any ion is going to bind into the channel, and they help in transfer of energy from one channel to another, this AMP, because some molecules does need energy and some does not need energy to cross the membrane, and the molecules which are impermeable to membrane, they need a carrier or a protein that helps them to cross the membrane, right? And they also, while crossing the membrane, they do need energy. So this CAM helps those kind of molecules pass through the membrane, right? And it also activates kinase protein. So this kinase protein, where we seen this kinase? Can anyone tell me? Yes, this kinase proteins we have seen in metabolic pathways like our glycolysis, cyclic or your Krebs cycle pathway, or cyclic AMP pathway, or electron ETC pathways. You can see this kinase. Mostly you can see this in glycolysis and Krebs cycle. Okay, and it also activates our calcium channels. As you know, calcium, you say a part, it helps in bone growth also in humans. Likewise, in other cells also, these calcium channels help in activation of growth hormones, especially in our humans, or you can say it all living beings, excluding all those bacteria and all, not those, but the animals and humans. So next question, what is genetic code? The genetic code is nothing but, as we know, every if we are doing some work or some we are playing any game, they have rules and regulations, right? Like that only our DNA have sets a few rules, and these rules to be followed in a sequence by the cells or the acids or the proteins that are involved in DNA replication and all. So the genetic code, it is a set of rules that determines how the nucleotide sequence of the DNA is translated. I'll tell you this in detail. in further, okay? For long answers, you can write it in detail. For small answers, you don't need to write that much in detail. So this gives a set of rules to be followed for the replication or translations of DNA and RNA, which are converted into amino acid proteins. Okay, it involves codons, which are 3-letter sequences of nucleotides that specify a particular amino acid to stop or start protein synthesis. Like, see, if you this code is a rule type. So it says when you have to start your genetic protein synthesis or when you have to stop it. Okay, next one. What is jaundice? And write its symptoms. So this jaundice, you can see whenever a patient who is suffering with jaundice, his skin or her skin completely becomes yellow, and his tissues, so the why his skin is becoming yellow, because of his tissues, cells, membranes are turning into yellow. Why? Because the bilirubin is secreted excess amounts in his blood, which is causing the cells to convert the color into yellow. Okay. It often indicates underlying liver or gallbladder problems. So if there is any misfunction in our gallbladder or liver, the person will be affected by jaundice, as the bilirubin is not secreted properly or excreted out of our body. It gets accumulated in our body and we get jaundice. How to identify the jaundice? One, our skin will become yellow, our eyeballs or our mucous membranes, they turn into yellow. This is one symptom. The other is fever, chills, severe abdominal pain, and he also have flu-like symptoms, little headache, and as mentioned you, change in skin color. The dark color of urine or the stool which has been excreted turns into blackish or brownish color like clay-like texture. Okay, so these are the symptoms of jaundice. So next, what is gout disease? Gout it is a type of arthritis. You know, most will be hearing from our adults saying, I got arthritis, I have no joint pains, knee pains. So how does they get arthritis? Or you can say it because the uric acid crystals which are formed, they get accumulated in the joints and tissues. So that's why, due to accumulation of this uric acid, it leads to severe pain. And you can say like something in a closed, you have a small particular space and everything gets dumped, it gets enlarged. So that is a container that is getting big and stuff. And we are humans, we have cells and pains. That's why they'll be getting pain, severe joint pain due to severe dumping of uric acid. The legs will become swollen as the uric acid bin, as you fill a balloon, take a filling water, it gets from this normal size to bigger size, right? Likewise, only the deposition of uric acid causes inflammation in our tissues and swelling. And it even occurs in not only joints of our knees, but also it can occur in toe area and the joints of our toes also. Define enzymes and right. So we have completed our 2 marks answers. Now let's move on to long answer questions, which are 10 marks. Okay. So here goes the question. Define enzymes. Write their IUB classification and factors that affect the enzyme action. So for any time when you're writing your 10 marks answer, if you think, even though whatever the topic may be, you have to just have a knowledge and a grasp of the topic and try to elaborate the answer. So don't try define enzymes as single line answer ho gaya, finished. And then IUB classification means the 5 classifications what we know, and just writing it, you say 10 marks that this much only answered. No, you have to try to elaborate your answers, and the elaboration will give you marks. So let's move on to the answer. Enzymes may be defined as biocatalytic catalyst which are synthesized by living cells. They are protein in nature. So there is an exception here. In RNA, that is also act as a ribozyme. Okay, so that is an exception. They're colloidal and thermolabile in character. They show this action in a specific manner. Okay, so mainly there are 2 types of enzymes, intracellular and extracellular. As the name indicates, intra means inside. So their function is within the cells where they are synthesized. Extracellular enzymes, extra, name indicates extra means outside. These cells are active outside the cells, and mostly example of your extracellular enzymes is digestive enzymes. So you can say it like if you, as we know, the secretion of the, for example, you take HCl acid that is secreted in the stomach. So if it is secreted, so that's why it is helping in digestion of our food. If it is the cells, if these enzymes are secreted in our inside the cells, how does the degradation or breaking down of our food happens, right? So digestive enzymes are an example of extracellular enzymes. So IUB, International Union of Biochemistry, have classified few enzymes into different categories. We know there's different kinds of enzymes, and you can't write each and one enzyme, right? So these what International Union have segregated the enzymes into different classifications in the year 1961. So what does this committee do? Does is this committee made a thorough study of the existing enzymes, as I mentioned you. It has studied all the existing enzymes and their principles, and based upon their action, it have placed them into different classifications and nomenclatures. And from 1964, the classification has come into action. And 1961, they made the plan, and 1964, they made the action. So here is the classification. It is classified into six parts. Okay, first is oxidoreductases. So what does the classification oxidoreductases does? You see oxidation, they catalyze the oxidation as well as the reduction reactions. Okay, and they transfer hydrogen and oxygen atoms or electrons from one substance to another. Let us see this reaction. See, for an example, we have given AH is one. You see one reaction, plus the another is another B is compound, and your H is the hydrogen. So as I'm saying, it is an oxidation or reduction. So what is happening here? It is getting reduced. The hydrogen that is present with A has been shifted, got reduced and combined with the B. So this is a reduction reaction. And how does it oxidizes? A molecule of A plus a molecule of O got combined and formed a single molecule. So here goes oxidation. What example of what are the example of oxidoreductases? Dehydrogenase and oxidase are the examples for oxidoreductases. Next classification is transferases. Transferases are these are functional groups which from one substance, they move to the other substance. These groups may be methyl, acyl, or amino or phosphate groups. These groups are only transferred. So that's why they are called as transferases. Here you can see A and B are molecules and C is a different molecule. Now what happened here? The B molecule got transferred to C molecule. Okay, so it is being like now A became a different molecule and BC became a different molecule. Example goes transaminase and kinase come to the classification of transferases. Next is your hydrolases. So what are hydrolases? These are these group of enzymes form two products by substituted. They form products from hydrolysis by substitute. Okay, so AB is a molecule, and you're adding like water molecule H2O, which give rise to here oxidation happens, and here AOH is hydrolyzed, and BH forms another molecule. So example is lipase, amylase, peptase comes under hydrolases. Next classification is lyases. These lyases are non-hydrolytic addition or removal of groups from substrates like carbon to carbon bond, carbon to nitrogen bond, or carbon to oxygen, or carbon to sulfur bonds. These bonds are cleaved. As you know, lyases means splitting. You can say it. So here is the example. RCOCOOH. This molecule is been reduced to RCOH plus CO2, or you can see the molecules has been split up, right? So lyases split up the carbon to carbon, or carbon to nitrogen, or carbon to oxygen, or carbon to sulfur bonds. So one of the example is decarboxylase. Next comes isomerases. These isomerases are intramolecular rearrangement. They do intramolecular means they do arrangement inside the same atom or same molecule. Likewise, AB in presence of isomerases, it forms BA. So here they did a rearrangement. Example, isomerase and mutase. Ligases means ligase. They combine the molecules. They join two molecules either by synthesizing it into a carbon to oxygen, or carbon to sulfur, or carbon to nitrogen, or carbon to carbon bond. They join these molecules with any of these, and they do a simultaneous breakdown of our ATP molecule. So let us see the example. Let us think X and Y are different molecules, two different molecules. Along with ATP, they are forming into a single molecule with an addition of ADP and PI. Your PI is nothing but phosphorus. So example of this enzyme is synthase. Moving on to next question. Write in detail about the conversion of cholesterol into vitamin D and bile acids. So this question is the most repeated question, I would say, and mostly focus studying on this question. It might come repeatedly for 10 marks. Okay. So how do you frame your answer? First, write about the definition of cholesterol, and then later on divide your answer into how it is the cholesterol is been converting into bile acids, or how it is converting into vitamin D. So let us go to the answer. Cholesterol. cholesterol nucleus, they are ring in structure, and these cholesterols, they cannot be metabolized. We can't ourselves metabolize, our cells cannot metabolize the cholesterol on their own. So what happens is cholesterol is 50% converted into bile acids, and it has been secreted or excreted in feces, or it is synthesized by two steroid hormones or vitamin D or coprostanol or cholestanol, and then later it is get excreted out. Cholesterol first letters come into synthesis of bile acids. So what happens is in the synthesis, the bile acids generally possess 24 carbon atoms and 2 or 3 hydroxyl groups in the steroid nucleus, and their side chain is ending in carboxyl group. Okay, they are amphetaminic in nature, as they possess both polar and non-polar groups. So that's why we are calling it as amphetaminic. So they serve as emulsifying agents in intestine and participate in digestion and absorption of lipids. So let us go to the steps. Okay. First is generally the synthesis of the synthesis of bile acids take place in liver, and it occurs in different steps. Okay. So the first step is the cholesterol is catalyzed by 7-dehydroxylase. So these bile acids inhibits this enzyme. So that's why we are calling it as rate limiting reaction. So the cholesterol in presence of this enzymes forms into two components, which is cholic acid and chenodeoxycholic acid. These are primary bile acids, and then these are found in large amounts in bile. So here you can see the synthesis. As I have told you, cholesterol is hydrolyzed first into 7-hydroxycholesterol and then forms into two components, which is our cholic acid as well as chenodeoxycholic acid. Now again, these two undergoes different reactions. So what happens is, on conjugation or in presence of glycine, this cholic acid converts into glycocholic acid, and in presence of taurine, the cholic acid converts to taurocholic acid, and again in presence of intestinal bacteria, glycocholic acid converts into deoxycholic acid. Okay, and then coming to chenodeoxycholic acid, in presence of either glycine or taurine, it converts into tauro or glycochenodeoxycholic acid. The same process, same reactions, but the difference is the end products of this synthesis are different. Okay. In presence of intestinal bacteria, it converts into cholic acid, and here you can see the cholic acid is ultimately converting into deoxycholic acid, and chenodeoxycholic acid is ultimately converting into lipocholic acid. Okay, as I have told you, on conjugation it happens, and the end products will be our deoxycholic acid and lipocholic acid. Next, after these two processes happening, then enterohepatic circulation will be occurring. So the conjugated bile salts, which are synthesized in our liver, gets accumulated in our gallbladder, and then from there they're secreted into our small intestine, where they act as emulsifying agents for digestion. and they absorb fats and fat soluble vitamins. So when large amount of bile salts are accumulated, they get reabsorbed uh, by liver through portal vein. So finally, these salts get again reused several times a day. So this is known as enterohepatic circulation. So next, let's move on to the conversion of cholesterol into vitamin D, or synthesis of cholesterol to vitamin D. So what happens is here, the 7-dehydroxycholesterol, which act as an intermediate for synthesizing cholesterol, okay, and then converting into our vitamin D. First, what happens? It first the cholesterol is converted into vitamin D3, which is cholecalciferol by in presence of UV rays, which means when UV rays fall on our skin, the cholesterol which is present, it gets converted into vitamin D3, which is cholecalciferol. So by absorbing this UV radiation, the 7-dehydroxy cholesterol turns into pre-vitamin D3, and then that pre-vitamin D3 gets converted into cholecalciferol, because that pre-vitamin D3 is unstable, and then ultimately it have to convert into cholecalciferol. So this, whatever this conversion of cholecalciferol is happening, that occurs in cellular membrane, and whatever the vitamin D is caused in cellular membrane, it gets expelled out to your extracellular space. And then, as you know, not only in the form of conversion, we get vitamin D not only through the conversion of the synthesis of a cholesterol to cholecalciferol, but through dietary take also you can take in the form of vitamin D3, which is fish and meat, food products. Okay, and also you can say the production of vitamin D3 in skin is the primary source where we get our vitamin D. And it also, if you take proper food, or like such as fish oil or mushrooms, these are in the form of D2, ergo calciferol. So the skin, whatever the synthesis it is doing to vitamin D3, it is due to presence of UV radiation. Okay, and then whenever the sun blockage is happening, only you get some proper sunlight on the high amount of UV light in mornings, and whenever you're not getting sunlight, what happens? It reduces. Finally, it gets reduces the sun blockage, and then people who are or people who doesn't receive good amount of sunlight, in like people who are living in higher altitudes, they don't get proper sunlight, right? So what happens is, for them, in our body only itself, our cholecalciferol thus is hydrolyzed by liver cells and forms 2,5-OHD, and then that 2,5-OHD finally ultimately converts into 1-alpha hydroxylation. Okay, it converts into 2,5-OHD by 1-alpha hydroxylation. So this is the active form. This is the active form that is produced in last step, and mainly in the kidneys and other renal sites, which the ultimate product will be 1,25-OH2D3. Okay, see, as I have told you, in presence of sunlight, our cholesterol will be converting into cholecalciferol, and whenever the sunlight is inactive, cholesterol becomes inactive, our liver takes it in as the whatever through direct intake we are taking, that liver converts it into 25 hydroxy vitamin D3, and that ultimately gets converted into 1,25 dihydroxy vitamin D3, which maintains calcium balance in our body. This is done in our kidneys. Okay. Next question. Explain the DNA replication process and enzymes involved in the process. So what is DNA replication process? Replication means nothing but duplication or multiplying. Okay, sorry. So what happens here is the DNA gets multiplied. Okay. So what happens, as you know, let's come from the starting. Okay. You know, the cells, whatever cells, in through meiosis, the parent cells produce daughter cells. Now, this DNA replication process occurs in our daughter cells. In the phase, in S phase of the cell cycle. So, whenever the daughter cell is in interphase, it has to replicate its DNA. So, DNA replication, you can also call it as DNA duplicating itself. So how does this process start means the DNA fragments, you know, we say our DNA is double helix, right? So what happens is it begins with unwinding our DNA helix, and each pair of the nucleotide will have a base pair of hydrogen bondings. So now our DNA will become split. Now it is not unstable, right? So we have to make it a stable, and to make it a two different DNAs, there are involvement of two different enzymes which help in replication process. You can say it, our DNA replication process as semi-conservative or conservative. Why do you say it's semi-conservative? Because the replication, what is happening, if it is having one parent bond in the one strand will be the parent, and the other strand will be a new formation, new strand of the DNA. You call it conservative when both strands are parent and the only two are parent and the other are all new strands. So here, as you know, general DNA, there's a basic base pairing occurs in for tyrosine, adenine on a new strand, and glycogen and cytosine together, they form base pairs. Okay. So this is how our DNA replication happens. This is our DNA. First, it gets split into two parts. It gets split. So, okay. What I told you that enzymes will be doing the splitting, right? So mainly, let us see how actually the DNA replication occurs. So this replication is very tightly controlled process. It does not occur very frequently. It occurs specific times during the cell cycle. So as I have told you, it occurs in interphase, or you can call it as an S phase of the cell cycle. So what does it need? It needs proteins and enzymes, and also it requires ATP because energy. As I told you before, when you go to your when I told you about 2 marks answers, the ATP helps in as an energy for processing, right? So here also we need energy. This occurs in 2 basic steps. First is initiation, and then elongation, and the other processes also. So this DNA replications occurs in both type of cells, prokaryotics as well as eukaryotics. Okay. So if you think like, I hope you guys know about video games, right? So if you think DNA replication is a kind of video games, you need few characters to play the game, right? So let us think our enzymes as characters and our replication is the game. So what kind of characters are here present for playing the game of the DNA is first you need DNA polymerase, which is your, there are also again DNA polymerase 1, 2, and 3, DNA helicase, and single strand binding proteins, primase, and ligase. So what happens is, as you can see here the strand of 5 to 3, here as you can see here will be the strand of 5 to 3. So whatever it is, helicase is like scissors, it splits the DNA to two halves. So splitting is done. Someone um, should do the replication, right? So we will think our DNA polymerase builder, because it builds our DNA or it builds the strands. So, but this DNA polymerase doesn't know where to start. So here primase comes the activation. What does a primate do? It tells this site where it need to start the process. So what happens here? Helicase splits the DNA, and our primase starts where where the binding should start, and that DNA polymerase bind to the primase and start its process of DNA replication. And here topoisomerase are also plays a role. What happens, what does they do? Means they stop the super coiling of our DNA. Means it should not coil, or it should not get into so clumsy or get what do you say more compact. So that's why here topoisomerase comes and stop and makes says or stops coiling of our DNA. So now you know our the basic DNA runs with 5 dash to 3 dash, again 3 to 5, right? So what happened is this this ligase, sorry, this primase starts with 5 and 5 dash and then ends with 3 dash. So what happened is whenever it is coiling with starts the process 5 dash to 3 dash, we call it initiation. So this is the first step. It is known as foundation. So this point when it where the primase initiates the process of binding. So it starts a point from 5 dash to 3 dash, and and also to the strand there are single stranded binding proteins which are binded to this strand, and they are like, they doesn't like to get binding to the other strands, so they will be separated. So as a result replication forks is formed. As I have mentioned you, topoisomerase prevents DNA from supercoiling, and now primase performs RNA performs the replications. So wherever the primase start, The DNA polymerase recognizes the movement and then start binding it. So your 5 to 3 dash will be your initiation, and then comes elongation. Here elongation, as the DNA are anti-parallel to each other, that is, as I told you, 3 dash to 5 dash and 5 to 3, these DNA polymerase 3 enzyme bind to the primer and it builds a new strand anti-parallel to it. For suppose 3 dash to 5 dash, and then it binds from, so it forms 5 dash to 3 dash, and likewise for the other strand too. So it forms a new base. So one of the strand will be our old one and the other one will be our new one. So this is what kind of polymerization? Semi-conservative. So whenever these binding happens, so whenever there's a discontinuous, this is very discontinuous, and there is a little gap between them, and there will be separate fragments, and these fragments you call it as Okazaki fragments. And what happens between these Okazaki fragments are binded by ligase. It acts as a glue type. It binds all the Okazaki fragments. Now comes the final step, termination. In termination, two identical double helix DNA molecules will be from the original DNA molecule. And this whole process is your semi-conservative, because each of the two copies contains one original and one new made strand. So as I have told you, I'll repeat this answer because this is a very important one, and always the replication start with origin of replication. And here is your DNA strand, which is the double helix one. So you can see it runs from 5 dash to 3 dash and 3 dash to 5 dash. As I have told you, helicates start unzipping or breaking down the DNA. So what happens here again? Primase comes here, binds here, and along with primase, DNA polymerase binds it. DNA polymerase binds, and then start forming its bond. And then there will be different. See here different. Okazaki fragments are formed, and there will be a gap between them, and then ligase comes and bind the bonds, and the topoisomerase stops from recoiling or supercoiling. So this all process occurs inside the nucleus for prokaryotics. So move on to next question. Write a note on lipid metabolism. Lipids are distributed into 3 important compartments in the body, that is liver, adipose tissue, and blood. The major part of lipid metabolism takes place in liver and adipose tissue. The blood works as a medium for transportation of these lipids. Okay. So tissues like cardiac and skeletal muscles required lipids and ketone bodies for their metabolism. And then major aspects of this lipid metabolisms are involved with fatty acid oxidations. This fatty acid oxidation produce energy, or the lipid synthesis, what you called as the lipogenesis, occurs. The lipid metabolism is closely connected to metabolism of carbohydrates, which are converted into fats. First step in lipid metabolism is hydrolysis of lipids in cytoplasm, which produce glycerol and fatty acids. these glycerols are metabolized to dihydroxyacetone phosphate, and dihydroxyacetone may be also converted to pyruvic acid through glycolysis. We know the pathway of glycolysis, right? There are several steps. Glycolysis and presence of ATP molecules, the energy molecules, ultimately converts into pyruvate, right? So here, this dihydroxyacetone may be also converted to pyruvic acid through glycolysis pathway. So in addition, what happens, it can also, this dihydroxyacetone can also be used in glyconeogenesis for formation of glucose 6 phosphate to glucose, which again help in energy. So the hydroxyacetone can also be converted to pyruvic acid, as I have told you. And these fatty acids again are oxidized by acetyl-CoA in mitochondria using fatty acid. And the acetyl-CoA then ultimately converts into ATP, carbon dioxide, and water using citric acid cycle and electron transport change. Fatty acids are synthesized from carbohydrates and are occasionally from proteins. The carbohydrates and proteins have been first catabolized into coenzyme A. Depending upon the energy requirements, what are the energy, what kind of energy they need, either in abundance or in low, the acetyl-CoA enters citric acid cycle, or it gets synthesized in fatty acids. And this process, you called it as lipogenesis. Next question. Describe various steps involved in glycolysis. These are all 5 marks answers, so you can reduce the size of your answer. As you know that lipid metabolism is a very lengthy answer where you have to explain detail about lipogenesis and the cycles involved in it, ketogenesis, lipogenesis, and all. These are very big pathways. So he asked for 5 marks. So we are writing a summary of all the cycles in your answer, as the questioner mentioned, note. So you are writing overall summary of the metabolism here. Okay. So as it is a 5 marks answer. So next comes our glycolysis. This glycolysis can be given as your long answer as well as your short answer. When keep in mind that whenever it is coming as a long answer, which is 10 marks, try to explain with its structures also, because as it is a 10 marks answers, you have to draw the structures. For a 5 marks answer, you don't need to draw structures. You can just write the flow chart. Okay. So let's get back to the answer. Glycolysis, as the term indicates, glycolysis, which indicates breakdown of glucose. Glyco means glucose. Lysis means breakdown. So glycolysis is the pathway which break down glucose molecules or glucose. So it degrades the glucose to pyruvic acid and energy to form adenosine triphosphate, which is our ATP, and reduced nicotinamide adenine dinucleotide, NADH. With the usage of ATP and NADH, our glucose is getting converted into pyruvate. Okay, this pathway occurs in all organisms, and most importantly, it does not require oxygen. Although it sometimes require maybe a little small, tiny amount of atmospheric oxygen may be needed. So let us see the equation which gives an explanation of series of reactions that are occurring in our glycolysis pathway. C6H12O6, which is our glucose, on addition with 2 NAD plus and 2 ADP with 2 molecules of phosphor give rise to 2 molecules of pyruvic acid with an addition of 2 ATP and 2 NADH and 2 molecules of hydrogen. There are several steps that are involved in glycolysis pathway, and these steps are divided into two main sections, which is your preparatory phase and payoff phase. First, let us study the preparatory phase. Preparatory phase is phosphorylation of glucose and its conversion to glyceraldehyde 3-phosphate. Okay, so the glucose gets converted to glyceraldehyde 3-phosphate. First, the glucose in presence of ATP converts to glucose 6-phosphate. This is the first priming reactions. Why do you call it a priming reactions? Because the ATP is converted into ADP. And this step, it is known as hexokinase. And the enzyme hexokinase helps the conversion of glucose to glucose 6-phosphate. And hexoisomerase helps in conversion of glucose 6-phosphate to fructose 6-phosphate. Okay. And the fructose 6-phosphate in presence of phosphofructokinase 1 converts to fructose 1,6 biphosphate. This is the second priming reaction. Why we are calling it as a priming reaction? As I told you, here ATP is getting released, okay, or it is getting converted into ADP. Next step, fructose 1,6 biphosphate is converting into glyceraldehyde 3-phosphate in the presence of aldolase enzyme. And here what happens is cleavage of 6-carbon sugar phosphate is happening to 3-carbon sugar phosphate. So as you can see here, the arrow marks, one is one side, and here you are having two arrow marks which indicate this is a single directional, and these two arrow marks which indicates bidirectional. So which means this reaction can be reversed. So glucose 6-phosphate can be converted to fructose 6-phosphate, and fructose can also be converted into glucose. And likewise, the glyceraldehyde 3-phosphate to fructose 1,6-phosphate can also be done. Okay. Here, you can see, as I have told you, glucose in presence of hexokinase converting to your glucose 6-phosphate, and isomerization reaction, glucose 6-phosphate to fructose 6-phosphate, and fructose 6-phosphate to fructose 1,6 biphosphate is converting, and then in phosphorylation, fructose 6-phosphate is converting to fructose 1,6 biphosphate, and here cleavage is occurring. Fructose 6-triphosphate is converting to two components, dihydroxyacetone phosphate as well as glyceraldehyde 3-phosphate. Okay, this is here cleavage is occurring. So next comes our payoff phase. Here payoff phase indicates whatever the compounds that are released in your preparatory phase are ultimately being used in our payoff phase. So what happens here? Oxidative conversion of glyceraldehyde 3-phosphate to pyruvate is been occurring from ATP and NADH. So glyceraldehyde 3-phosphate can be on oxidation and phosphorylation gets converted into 1,3 by phosphoglycerate, and the enzyme that is involved is glyceraldehyde 3-phosphate dehydrogenase. And here you can see, in presence of 2 molecules of phosphor and 2 NAD plus, here you are getting NADH and hydrogen. Here, following the next step, 1,3 biphosphoglycerate is converted to 3-phosphoglycerate in presence of phosphoglycerate kinase. The first ADP, ATP is formed here. And that too, this is also called as substrate level phosphorylation, where you have seen in preparatory phase, the ATP is converting into ADP. Here ADP is converting into ATP, and you get 2 molecules of ATP here. 3-phosphoglycerate is converted to 2-phosphoglycerate in presence of phosphoglycerate mutase. And 2-phosphoglycerate converts to phosphoenol pyruvate in presence of enolase, and here water molecule gets released. Okay, here I'll show you, see the structure of it. Here you have an OH. Here, okay, H2O water molecule is present in your second position, and here the water molecule left. So what is happening here? Dehydrogenation reaction is happening. Okay, and the final from phosphoenol pyruvate to pyruvate is been formed in presence of pyruvate kinase, and here our second ATP is being formed. So overall, in glycolysis, 4 ATP molecules are being formed ultimately. Okay. So here are the following reactions of the same, but here in a simple form. So you get an idea of it. So move on to next question. Write the synthesis and significance of biological adrenaline. Okay. Adrenal medulla is an extension of sympathetic nervous system. So it produces mainly 2 important hormones, which are epinephrine, or you can call it as adrenaline, or norepinephrine, you can call it, or noradrenaline. These are the 2 important hormones that are being secreted by adrenal medulla. These hormones comes under catecholamines, as they are amine derivatives of catechol nucleus. This catechol nucleus is nothing but dihydroxylated phenyl ring. So what happens here is, the epinephrine is a methyl derivative of norepinephrine, which means adrenaline is a methyl derivative of noradrenaline. And dopamine is also another catecholamine which is formed during the synthesis of your epinephrine, and it is and this dopamine is an intermediate. It acts as an intermediate. Okay. The norepinephrine and dopamine are the main and important neurotransmitters in our brain and help and in our autonomous nervous system. So as I told you, they are derivatives of catechol. What the name catechol refers to dihydroxylated phenyl ring. The phenyl ring, you call it as a catechol. The amine derivatives of this catechol you are calling as catecholamines, means dihydroxylated amines of the phenyl rings are your catecholamines. So thyrosin is the main enzyme that is helping for your synthesis of catecholamines. namely dopamine, norepinephrine, and epinephrine. This tyrosine is the main enzyme that helps in synthesis of your dopamine, norepinephrine, and epinephrine. So let's see how the synthesis occurs. So your L-tyrosine. So here goes your first step. Hydrolysis to 3-hydroxyphenylalanine, or you call it L-dopa. Levotyrosine. L indicates levo position. Okay. So in presence of tyrosine hydroxylase forms 3-dihydroxyphenylalanine, and this L-dopa in presence of L-aromatic amino acid decarboxylase converts into dopamine. This dopamine in presence of dopamine beta hydroxylase converts to norepinephrine, and the norepinephrine converts ultimately to epinephrine in presence of phenylethanolamine and methyl transferase. So this is how your epinephrine gets synthesized from tyrosine to epinephrine. Okay. The matter here present is the same what I have explained you. So let's see what are the functions of catecholamines. So what does these catecholamines do? Either it be epinephrine or norepinephrine, they regulate the carbohydrate and lipid metabolism. They stimulate the degradation of triacyl glycerol and glycogen, and they also increase our blood pressure. And these dopamine and norepinephrine, as I told you before, they act as a neurotransmitters in our brain and autonomous nervous system. So these two hormones or enzymes, they help in transferring of nerve signals. Okay. So next question, explain oxidative phosphorylation and its mechanism. So what is oxidative phosphorylation? So it is a process where ATP is synthesized using energy that has been generated by electron transport chain and proton gradient. The oxidative phosphorylation, it is a final step of our cellular respiration. So overall, the end cycle of our cellular respiration is our oxidative phosphorylation. So it generally occurs in mitochondria, and it is linked with the process of electron transport chain. The electron transport system is located in inner mitochondrial membrane. The electrons are transported from one membrane to another transport chain, and then it goes a series of redox reactions. Okay. So how does the electrons get delivered by NADH and FADH? So the electrons be delivered by, we have seen in the glycolysis, the reactions that occurs in between NAD and getting converted to NADH. Likewise, these NADH and FADH helps in conversion or delivery of the enzymes. Okay. So NADH and FADH2 are reduced and transfer their electrons to molecules that are near the beginning of the electron change. After transferring the electrons, they get oxidized to NAD plus and FAD. And these are utilized in other steps of cellular respiration. So next is our electron transport and proton pump. So electrons move from high energy level to low energy level. Thereby, while they're moving from high energy to low energy, they release some energy. Okay. that energy is used for moving the electrons from the matrix into the intermembrane space. So this electrochemical, so when it is happening, the electron movement of the electrons, electrochemical gradient is produced at that time. So now next, what happens is the oxygen is split to form water. So electrons are then transferred to oxygen molecule, and then this oxygen molecules get split into half. It uptakes hydrogens and forms water. So it takes two hydrogen molecules and then converted into water molecules. Now next step is our ATP synthesis. Here the hydrogen ions passes through the enzyme called ATP synthase when they're coming back from the matrix. And then this controls the flow of proteins to synthesize the ATP. Okay, these ions when they're flowing come back in front, it controls the synthesis of the ATP molecule. Next, chemiosmosis. Chemiosmosis, which may indicates it is a oxidative phosphorylation which uses chemical reactions that release energy to give a chemical reaction that requires an energy. These two sets of reactions are coupled and interrelated. The electrons that flow through electron transport chain is an exogenic process. And the synthesis of the ATP is an endogenic process. These two processes are ingrained within the membrane itself. And ultimately, the energy will be transmitted from electron transport chain to ATP synthase by the movement of the proteins. This process is termed as chemiosmosis. The endogenic process is a chemical reaction in which energy is absorbed. So there will be a change in free energy, and this energy change will be positive. Exergonic process is also involved, which is a chemical reaction in which there will be positive flow of energy from the system to the surroundings. The chemical reactions are also considered exergonic when they're spontaneous. Next, electron transport chain. So most of the biochemical catabolic processes like citric acid cycle, glycolysis, beta oxidation, etc., which produce the NADH coenzyme, they consist of electrons having high transfer potential. So these reactions release abundance of energy on oxidation. These reactions are also uncontrollable as they are releasing so much of energy within the cells, and they are not releasing all at once. They're continuously just little amount of energy they're releasing. So these electrons are then separated from NADH and then passed to oxygen with a series of enzymes, releasing small amount of energy, as I have mentioned you. All the series of enzymes having complexes. They also have complexes for electron transport chain. These complexes together you call it as an electron transport chain. This chain can be seen in inner layer of the membrane of mitochondria. The salts of succinic acid are oxidized by this electron transport chain system. In eukaryotes, these enzymes make use of energy that can be released in electron transport system from the oxidation to NADH. that pumps protons across the membrane. This can be considered as one of the best examples to understand the concept of oxidative phosphorylation. So these are the different types of pathways or the cycles that are involved in your oxidative phosphorylation. So the answer is for 5 marks. So you can cut short the answer, as you can't write whole cycles. You can't describe the TCA cycle all at once, because that would be a 10 marks answer, right? So you have to take the main points of the each cycle and write it in your answer if they're asking for 5 marks. Next question. Explain the urea cycle and its disorders. So urea cycle is also known as ornithine cycle. It occurs a series of biochemical reactions that occur in our liver and some other tissues to eliminate excess nitrogen in the form of urea. It plays crucial role in body's nitrogen metabolism, and it converts toxic ammonia into less toxic urea, which can safely get excreted by the kidneys. The urea cycle consists of several interconnected steps and each catalyzed by one specific enzyme. So here is a picture of your urea cycle. So in your first step, the carbamyl phosphate formation occurs. So what happens is the cycle begins in the mitochondria of our liver cells with the conversion of ammonia and bicarbonate into carbamyl phosphate. This is catalyzed by the enzyme carbamyl phosphate synthase 1. In short, CPS1. So next step will be citrulline synthesis. This CPS1 combines with ornithine from citrulline and then takes place in mitochondria. This combination occurs in mitochondria and catalyzed by ornithine transcarbamylase enzyme. Here comes your second enzyme. And then in third step, aspartate incorporation occurs. The citrulline is transported from mitochondria into cytoplasmic of liver cells. In the cytoplasm, citrulline reacts with aspartate to form arginosuccinate. This is catalyzed by arginosuccinate synthase. Next, the cleavage of arginosuccinate, the splitting of arginosuccinate. The arginosuccinate is then cleaved or splitted into fumarate and arginine by enzyme arginosuccinate lyase. The fifth step is arginine, whatever is formed, is then converted to ornithine. The arginine gets hydrolyzed from the urea and ornithine. The enzyme that is causing this hydrolation is arginase. Then ornithine renally re-entry into mitochondria. Again, this ornithine what is formed is again re-entered into mitochondria and then participates in another urea cycle. Next, regeneration of fumarate. The fumarate gets regenerated during the splitting of arginosuccinate and then gets converted back to aspartate in citric acid cycle and then again re-enters into urea cycle to combine with citrulline. And then whatever the urea that is formed, it's get final product that gets excreted. The excretion, the urea that is the final product of the urea cycle, it is released into the bloodstream and filtered by kidneys for excretion in urine. This is involved by different factors like dietary protein intake, how much proper diet you take or you consume it, and then how much amount of ammonia is toxicity occurring in your body, and these conditions you call it as urea cycle disorders. As you can see, the accumulation of the urea, abundance accumulation of urea in your body or yourself, that will cause to life threatening and may require intervention, or sometimes you may it leads to death too. So overall, this urea cycle plays some important role and maintain nitrogen balance in our body by converting ammonia into urea, which can be safely excreted by preventing ammonia toxicity. So here are the disorders that can be occurring due to accumulation of urea in our body. So if there is a deficiency of N-acetyl glutamate synthase, then you will be suffering with hyperammonia that may be accompanied by high plasmic concentration of alanine and glutathione. What are the clinical features? You see, lethargy means persistent vomiting, poor feeding, hyperventilation, enlarged liver, and seizures. The deficiency of carbamyl phosphate synthase causes citrullinemia, respiratory alkalosis, hyperammonia, lethargy is your clinical same, and coma. You may get seizures, vomiting, poor feeding, hyperventilation, hepatomegaly. And next, the deficiency of ornithine transcarbamylase leads to respiratory alkalosis and elevated orotic acid in urine. Again, the symptoms will be vomiting, hyperventilation, hepatomegaly. And the deficiency of arginosuccinate synthase causes citrullinemia. The symptoms will be same: seizures, vomiting, poor feeding, lethargy, hepatomegaly. The deficiency, another deficiency is arginosuccinate ligase. The deficiency of this enzyme may lead to elevated arginosuccinic acid in urine. The symptoms will be the same: vomiting, poor feeding, hyperventilation. Next, the deficiency of arginase will lead to elevated plasma argine, lactate, and CSF glutamine, and elevated blood ammonia. The symptoms will be loss of muscle control, seizures, irritability, delay in development, and protein intolerance. These are the disorders of your urea accumulation, okay, in the body. Next, moving on to next question. Explain citric acid pathway. Citric acid cycle, you can also call it as Krebs cycle or carboxylic acid cycle. It is the most important metabolic pathway for the energy supply to the body. About 65 to 70% of the ATP is synthesized in Krebs cycle. The citric acid involves the oxidation of acetyl-CoA to carbon dioxide and water. This utilizes 2/3 of the total oxygen to consume by the body. The name TCA is used since the outset of the cycle, means the end products of the cycle are tricarboxylic acids, citrate, cis aconitate and isocitrate. Okay, so let us see the pathway and the steps involved in it. First is formation of citrate. It starts with condensation of acetyl-CoA and oxaloacetate, which is catalyzed by citrate synthase. And second and third steps, it is citrate is isomerized into isocitrate by enzymes aconitase. And then cis aconitase is converted to isocitrate into aconitase. Again, here water molecule is utilized, and isocitrate converts to oxalosuccinate in presence of isocitrate dehydrogenase, and oxalosuccinate gets converted into alpha ketoglutarate into by oxalosuccinate decarboxylase enzymes, and here carbon dioxide is released. Here CoA is converted into carbon dioxide, and in presence of NADH, in presence of alpha ketoglutarate dehydrogenase, it is converted into succinyl CoA. And the succinyl CoA gets converted into succinate in presence of succinyl CoA synthase. Here you get GTP and coenzyme A. Succinate gets converted into fumarate in presence of succinate dehydrogenase, and then fumarate converts into maltase in presence of fumarase. Maltase gets converted to oxaloacetate in presence of malate dehydrogenase, and then again the cycle gets repeated. Oxaloacetate gets converted into citrate in presence of citrate synthase. The cycle goes on repeating, and there are 10 steps which are involved in this cycle. Okay. Next question. Explain the structure of coenzymes and their biochemical functions. The non-protein, organic, low molecular weight and digestible substance associated with enzyme function is known as a coenzyme. They are often regarded as second substrates or co-substrates, since they have affinity with enzymes that are compared to substrates. They undergo alternations during enzymatic reactions and then later gets regenerated. This occurs due to contrast to the substrate, which is converted into the product. They are often derived from vitamins and can be loosely bonding to and loosely bonded to. Example, for example, NAD+. NAD+ and also heme prosthetic group of cytochrome C to enzyme. This is also loosely bound. The tightly bound coenzymes are called prosthetic groups. Loosely bound coenzymes are like substrates, that is, they bind and release to enzyme just like the substrate and products. The functional enzyme is also referred as oleoenzyme, which is made up of protein part. The protein part is apoenzyme and the non-protein part is coenzyme. So coenzymes form B complex vitamins. The most of the coenzymes are the derivatives of water soluble B complex vitamins. That means biochemical functions of B complex vitamins are exerted through their respective coenzymes. Non-vitamin coenzymes means they are not at all coenzymes. These are not derived from vitamin derivatives. Some substances have no relation to the vitamins but function as coenzymes. They considered as non-vitamin coenzymes. Example, ATP, CDP, UDP, etc. Nucleotide coenzymes. These type of coenzymes possess nitrogenous bases like sugar and phosphate. These coenzymes are therefore regarded as nucleotides. Example, NAD plus NADP plus FMN, FAD, coenzyme A, UDPG, etc. These are nucleotide coenzymes. Protein coenzymes. Thioredoxin, it is a protein that serves as a coenzyme for the enzyme ribonucleotide reductase. Coenzymes do not decide enzyme specificity. So a particular coenzyme may participate in catalytic reactions along with different enzymes. For example, NADP plus act as a coenzyme for lactic dehydrogenase and alcohol dehydrogenase. In both the reactions, NADP plus is involved in hydrogen transfer. The specificity of the enzyme is dependent on apoenzyme but not on coenzyme. Next question. Explain the electron transport chain. The ETC, it is located in inner mitochondrial membrane of eukaryotic cells or plasma membrane of prokaryotic cells. So it is a series of protein complexes and coenzymes that work together to transfer electrons and pump protons across the membrane, creating an electrochemical gradients. So the steps which are involved are, first step is complex 1 NADH Q oxidoreductase. So it compromises the enzymes consisting iron, sulfur and FMN. Here what happens is the two electrons are carried to first complex 1 and about NADH, MN, FMN, and these are derived from vitamin B2. Second step is Q and complex 2 succinate Q reductase. In this step, FADH is not passed through complex 1 and which is passed, it is received directly from complex 2. The first and the second complexes are connected to a third complex through compound ubiquinone, your Q, that is Q. This ubiquinone is soluble in water and moves freely to the hydrophobic core of the membrane. In this phase, the electron is delivered directly to the ET's electron protein change. The number of ATP obtained at this stage is directly proportional to the number of protons that are pumped across the membrane, the inner membrane of the mitochondria. Okay. Next step is complex 3, which is cytochrome C reductase. The third complex is comprised of iron to F protein, cytochrome B and cytochrome C proteins. Cytochrome proteins consist of heme group. The complex 3 is responsible for pumping protons across the membrane. It also passes electrons to the cytochrome C, where it is transported to the fourth complex. From the third complex to it is transported to the fourth complex of the enzymes and proteins. So the Q electron will be the donor and the cytochrome C will be the acceptor here. Complex 4 is your next step. Here cytochrome C oxidase. The fourth complex is comprised of cytochrome C, A and A3. There are two heme groups where each of the item present in cytochrome C and A3. The cytochromes are responsible for holding oxygen molecule between the copper and iron until the oxygen content is reduced completely. In this phase, the reduced oxygen picks two hydrogen ions from the surroundings and form to a water molecule. You can see here in this picture, here is your matrix, inner membrane, and then interspace. The NADH plus here, H in complex 1 gets releases 3 hydrogen ions, and then the FMN here present is taken up by ubiquinone, and then from complex 2 succinate and fumarase act here, the FAD, and then gets again the FAD is released. The FAD is taken up by the ubiquinone, and then which passes to the complex 3, and here complex 3 4 hydrogen ions are released, and then site B is converted to site 1, which is ultimately converted into site C, and then site C binds the intermembrane space, and then in complex 4 it converts to site A and A3, where 2 hydrogen molecules are released here, and water molecules, the oxygen takes up the utilize the hydrogen molecules and forms water molecule, and the hydrogens which are hydrogens which are released out are taken up by ATP synthase, and then they are used. Okay. So here is an other question, which is the biosynthesis of pyrimidine nucleotide. So the biosynthesis of pyrimidine nucleotide involves the series of enzymatic reactions that occur in cells to create building blocks of DNA and RNA. First is your carbamyl phosphate synthesis. In this, pathway starts with conversion of glutamine, ATP, and biocarbonate into carbamyl phosphates. This is catalyzed by carbamyl phosphate. And next is your aspartate transcarbamylase. There, carbamylase phosphate reacts with aspartate, and then in presence of enzyme aspartate transcarbamylase to form carbamyl aspartase. Next is dihydroorotase. Carbonyl aspartase, which is formed, is then converted to dihydroxyorotase by dihydroxyorotase enzyme. The dihydroxyorotase is converted to dehydrogenase. Is then dihydroorotase oxidizes into orotate by dehydroxyorotate dehydrogenase by using NADE plus as a cofactor. Next is orotate phosphoriboxy transferase. The orotate gets activated by binding it to ribose 5-phosphate molecule, forming orotate monophosphate, and this is catalyzed by orotate phosphoribotransferase. And then orotyne 5-phosphate decarboxylated by orotyne 5-phosphodecarboxylate, producing uridine monophosphate. Next occurs UDP synthase. UMP is uridine monophosphate is phosphorylated to uridine diphosphate by help of UDP synthase. Here, in the next following step, UDP can be further phosphorylated to uridine triphosphate by UTP synthase, which uses ATP as substrate. These newly synthesized pyrimidine nucleotides, specifically UTP, are used as building blocks for RNA synthesis. To form the building blocks of RNA, enzymes like ribonucleotide reductase are responsible for converting UTP into deoxyuridine triphosphate, which is then further modified to produce the other deoxyribonucleotides such as deoxyadenosine triphosphate, DTAP, DCTP, deoxycytidine triphosphate, and DGCTP, deoxyguanosine triphosphate. And this process ensures that cells have constant supply of pyrimidine nucleotides necessary for DNA and RNA synthesis, allowing for the replication and growth of the genetic material. This is the biosynthesis of pyrimidine nucleotides. So thank you guys for staying up till the end and listening to this session. I hope you enjoyed this session, and I would like you all to follow and subscribe Prakara. Here in Prakara, we are a app. Prakara, we are an app which help you B Pharmacy students for such interesting contents, and we provide you notes, detailed explanation videos of each topics. We provide you keywords and MCQs for you people. So here we have now SEM 1 and SEM 2 notes, and we are having for curriculum MLP now, which is mini learning program. We have MLP mini learning program, which is for just 149 rupees only, and this will be you'll be given access for 21 long days for video lectures, student notes, key terms which are helpful for terminology knowledge. And you can also assess yourself after watching the videos, whether you have understood the topic or where you have missed your topic by doing the MCQs on our app. And please don't forget to subscribe Prakara for more such content. And if you have any queries, please do call to the number presented here. That is 741-6600-658. And I want you all to get this program, and I assure you this will be beneficial for you all. We have all notes from first year to fourth year, that is from first SEM to eighth SEM, and at now we are giving you an offer for rupees 149 only, and this is a very limited offer, and I hope you will grab this opportunity and do not miss this opportunity. For this 149 rupees, you'll be this trial pack. You can use, this is the trial pack, and you can use it for 21 long days, and I guarantee you that you will not be disappointed for joining Prakara.